the liver. The liver is the largest internal organ of the body and located in the right upper quadrant, outlined by the red circle here. And also you can see how it extends in the posterior region in this netter plate. Anatomically, the liver is divided into a very large right lobe, separated by connective tissue from the left lobe. If you turn the liver and view its undersurface, you see the caudate lobe in the posterior aspect, and this is the anterior aspect. The fourth lobe is the quadrate lobe. The green structure is the gallbladder, and we'll take a look at the gallbladder in lecture. It is attached to the liver by an adventitia. This structure here is a remnant of the umbilical vein. This is the ligamentum teres. In order to understand the function of the liver, it's important to consider the blood flow through the liver. In the digestive system, we talked about the first capillary bed being in the loops of intestine. The first capillary bed drains into a very large vein called the portal vein, which is carrying the majority of blood into the liver. The second capillary bed of this portal system is the sinusoidal capillary bed found within the liver. This netter plate nicely outlines the flow of blood in through the portal vein. This is primarily deoxygenated blood. Again, most of the oxygen has been extracted in the digestive system. This is the hepatic artery, bringing in the remainder of the blood volume. Approximately 25% of the blood volume is highly oxygenated blood. This is a branch directly off of the abdominal aorta. Note the green structure and the arrow, the direction of the arrow. This indicates the flow of bile out of the liver. This area that I'm circling here indicates a structure called the portal triad or the portal canal, and these are where the large vessels terminate. Looking at the microanatomy of the liver, we see that the square, or actually more, they're not considered to be cuboidal shaped cells here. They have more than 4 sides very often. These cells here are the hepatocytes. They are arranged in typically single layer plates. So single layer plates of cells surrounded by the second capillary bed, which is the sinusoid or the liver sinusoid. This structure here is the portal vein within that portal canal, also called portal triad. Here's the terminal branch of the hepatic artery, and this is the bile duct. So you can see from this illustration that the flow of blood is flowing from the region of the portal canal towards a structure called the central vein. So through the sinusoids, and then enters a venule called the central vein. This slide illustrates what you will see in lab on Tuesday. So you note the direction of the arrow, the flow of blood through the sinusoids of the liver, nourishing those hepatocytes and then draining towards the central vein. Hepatocytes produce and secrete bile into a duct system. The flow of bile is opposite of the flow of blood, as seen in the cartoon illustration on the left. Now, the microarchitecture of the liver is typically described in 3 organizations. The first and most commonly used term is the classic liver lobule. The classic liver lobule is seen here in the dotted line. We have so I'm drawing a circle around the classic liver lobule. The classic liver lobule has portal triads or canals on the periphery, and a central vein in the center of the lobule. The function of this organization is to note the flow of blood, again, from that portal canal region towards the central vein, and the flow of bile in the opposite direction towards the portal canal, as it is made by the hepatocytes and enters larger and larger ducts until it finally reaches the bile duct. The next video will describe the flow of bile through the liver. So again, this is the most commonly used term to describe the microarchitecture of the liver. So a central vein surrounded by portal canals in the periphery. Typically, you'll see anywhere from 4 to 6 portal canals in the periphery. Now, the second most commonly used organizational term is the hepatic acinus, and this is describing the flow of blood between two portal triads towards the central veins, two central veins. So you just typically draw a diamond structure where we see the access between two portal triads, again where the terminal vessels are found, both the terminal portal vein and the terminal hepatic artery, and the direction of the flow of blood towards two central veins. So we have an axis going in this direction as well. And then that area within this blue dotted diamond is divided into zones. So we have zone 1, closest to the portal canals. Zone 2 is considered to be the intermediate zone. And then zone 3 here is the zone closest to the central veins. Less commonly used is the description of the portal lobule. This is a triangular shaped organization where the portal lobule centers on a portal canal seen here. So 3 central veins on the periphery, central vein, and a central vein. And then again, at the center of that triangle is the portal canal. This organization is describing the exocrine function of the liver, which is the production of bile into a bile duct system that then exits the liver. So we see the production of bile by hepatocytes, increasing thickness and amount of our duct system as we lead to the portal canal. Okay, so just to review the microarchitecture, the 3 areas described when looking at the histological appearance of the liver. First, we have the classic liver lobule, which is describing the flow of blood from the portal canals towards the central vein. The hepatic acinus is describing the flow of blood between 2 portal triads towards 2 central veins, and that area is divided into specific zones. And the last and least often used is the portal lobule, which describes the exocrine function of the liver and the flow of bile from hepatocytes towards the portal canal where the bile duct is located. You can nicely see the bile duct in this illustration based on its cuboidal epithelium. So here's a profile of the bile duct, and here's a profile of the bile duct.